Today I have planned to address an issue of vaccination, which is primarily been discussed at length yesterday by a number of speakers, talking about the need of type of viruses or organism, characterizing them, typing them, and making them in a position, in a format which can be used in a vaccine more effectively. So I have just decided to discuss or present the concept of precision immunization. How can we make the immunization more precise? So this will be basically a kind of general information, but focusing mostly on the viruses which we have handled in Pakistan in poultry for the past number of years. So I'll try to make those examples and discuss it. Generally, for a few definitions for students or somebody who is not aware with the immunization concept, I know everybody knows what vaccines are. And then there are different types of vaccines. You must have heard since yesterday, a number of people have described it. And I think the main purpose is using why we use vaccine is to improve the health and productivity of animal, or you can say poultry particularly, which we are discussing today. The concept of precise immunization is that we have to characterize vaccine in a way that we understand the qualities of the host and the type of the organism which we are using, and then match it in a way that the best results could be obtained. That's kind of concept. Traditionally, vaccines are made by certain using the organism as a whole, but now with the introduction of biotechnology tools, it has become possible to specifically identify those components of the organism which are more important to generate an immune response. So we defined it as a homologous vaccine, in which we are using the precise organism which is available in the field, and you develop vaccine against those organisms. Rather than without knowing the organism in detail, molecular level, you have a vaccine. We call those vaccines which have not been designed to that parameter as heterologous vaccines. Generally, the concept is that how basically well a vaccine can stimulate immune response that we define it as potency of the vaccine. And that again depends upon how closely the antigenic mass or the type of antigen is basically matching with the vaccine, the match between the vaccine strain of the organism and the organism in the field, how closely they match. So that they will determine the antigenic match of the organism as far as vaccine is concerned. And then as a potency is influenced by these factors like strain quality, strain typing, adjuvant which we use, the formulation which we design. So all other factors do include in that one. But because I said earlier, my focus is primarily to talk about the effect or impact of antigenic variability and the impact of those antigenic characteristics on the quality of the vaccine. Generally speaking, basically, vaccines are prepared from the organisms which are, you can say, in the same group, serologically sometimes the same, but they're not identical, you can say, with the field organism. So in that situation, many vaccines do not respond, or do not perform the way we expect them to perform. So basically, the best thing will be either to test its titers against those antigens from the field virus, or challenge that vaccine with the field strain, and then qualify those vaccines to be more effective. It's kind of some definitions. So let's focus on the identification of those particular organism, the antigenic, the properties of the organism. And as we know, over a period of time, organisms do mutate. Most of the organisms, they mutate. They are from the RNA group of organisms. They are very fast mutating. You must have known about influenza, IBV. I'm talking about poultry perspective. And there's a good example of this livestock foot and mouth virus. So there's the number of serotypes, and then the number of subtypes, genotypes. The kind of complex of issues are there with those organisms. So it is very important to understand that you use the vaccine, how much effective that vaccine has been, That will, you only will know once it's applied in the field, and then you see the response after vaccinating. Do we have outbreak of the same organism or not? So that will qualify that vaccine effectively. Now, with the current information about from the molecular perspective about the structure of the organism, the DNA, stuff. So we are focusing more to characterize those organisms on the basis of their molecular characteristics, and then decide, this this character, which isolate, which strain, which subtype, which genotype would you like to use as a vaccine. And how it's important for the control of diseases, that's another part, because in the later presentation, I will give you some examples how we have tried it in the field. We know that serologically they're distinct. You cannot have protection using H5 vaccine against H7, and vice versa. And then there's some serotypes within IV viruses. I'm saying these things for students primarily who are less aware of it. So please bear with me. I know it's basic, for many of you who are experts, but we need to have background information. So in the field, you will see one vaccine claiming that it covers all serotypes. No problem. So these are all, marketing, tactics. But practically, scientifically, we have to be very clear that there's no cross protection between serotypes. Most of serotypes, they are distinct. And that makes the life of the manufacturer of vaccine difficult. Because personally, I'm also involved in vaccine production in Pakistan. So I think my life has become very difficult. After knowing this information, claiming it, preaching it, and then developing a product which basically covers those parts in talking. So the main responsibility goes to the vaccine manufacturer so that they can produce a fit for purpose vaccine for different diseases. So how we do it, you have to basically isolate the virus or bacteria, characterize it. The technique called cartography, you must have seen yesterday, Dr. Munir's, both Munir's, they have very explicitly explained their, this topic in detail, that how this cartography technique works. I will show you some pictures as well. And then we identify those epitopes of the antigen, which are basically desirable and more immunogenic. Sequence information also help us to define which protein structure they will express and how that protein will be immunogenic. Second thing to test is, after knowing all that structure of the organism, will be the response. And that's, as I said earlier, that's measured by the post-vaccine response. And post-vaccine response basically, again, will depend which antigen you have used to test that vaccine. So it's not only you're concerned with the vaccine development, you're also concerned to see the immune response. So if you're not using the matching antigen for the titers, you will have a different thing. This is just an example of a previous work we did when I was young, I can say, studying this H7 influenza. So we had this problem of avian influenza virus in Pakistan, H7 serotype in 1995. And then next time we had a problem in 2001 and 4. So we had this cartography at that time. We decided whether we are going to use the first virus we isolated in 1995 and use it as a vaccine, or we're going to use this new one. So this study clearly indicates that there are two distinct groups of those viruses. We could not have used the old virus. I'm talking about 2001. Now, after that, a lot of science has been added to this information. So you can see how much knowledgeable we are to prepare precise vaccine and develop those specific vaccines. Another example is H9. People are quite familiar because Pakistan is literally free from H5, H7, and we must still have H9. So similar things were done with H9, and the virus, the vaccine which we developed in those days was selected on that basis. Cartography. This I have borrowed from Dr. Munir's work because I said Pakistan is free from H5. So I didn't have much data to give you an example. So I requested him. It's his slide, and you can see categorically within H5, there are a number of subgroups and number of genotypes. And so this cartography indicates to you the distance between two organisms. If they are closely located, maybe you can have a close match. If they are distinctly related, you cannot use one for the control of the other. Now, how we can have this concept of homologous vaccine development? So, a number of things can be considered, like for live vaccine, for production of live tenor vaccines. What will be the approach? Basically, as the live vaccines are considered to have more prolonged immunity and better CMI response, so obviously we would like to develop those vaccines using those new isolates. The currently used vaccines, live vaccine, attenuated vaccines are developed in a different way. Some are natural organisms which are used as a vaccine. Some have been adapted. But with the new technique, like with the new technology, we can really clone it, develop it, So live vaccine can be developed. Maybe take a little more time than the killed one, but that could be approach once you characterize and see what kind of virus you are basically handling. The only problem with it was risk of virulence, risk of reversion of that virus. But it stands there still. But with the cloned, viruses are cloned, seed virus, there are some other problems, not primarily reversion, but that's kind of part of the live vaccine development. Other part is the inactivated vaccine, which you use is quite relatively easier, I would say, developing such a vaccine, provided you have information about the characteristics of the virus, grow it, the host, you know the cell culture, you would use egg, how would you use it? So that's the technique using it. But to make a homologous, you have to have the similar viruses close to the one which you are seeing in the field. And recombinant vector-based vaccines, still the vaccine currently used HPT vector, NDV vector are made, but they use the F gene from the Lesotho strain, for example. That's again same standard virus being used for many years. And we don't get into detail over here, time is short, that there's one serotype of this NDV. Why can't we have, the same Lesotho over there? I think I remember Dr. Munish saying yesterday, perhaps 25 genotypes mentioned about NDV yesterday, Dr. Munish. 21, yeah, 21. I thought I heard it. So there's a reason for doing it. So this technique, which was explained yesterday, can be used as a vector-based vaccine. We can develop very specific, precise vaccines. Then actually subunit vaccine. There are people in the room, professors, who have been doing work on this subunit vaccines. And Dr. Moaz, Professor Moaz is sitting here. He will do his presentation on adenovirus, I guess. He will explain you more about it. But that can be used, technique can be used to develop any new virus, may it be pathogenic, whatever, rather than using other approaches. This approach can be used. Again, this is borrowed from Dr. Munish's permission because we don't have a data with us where we have ourselves developed such a vaccine in which we can use this as a vector, virus, NDV used as a vector, and you clone the antigen of your interest into that. So this you must have seen yesterday as well. But this explains how easy, I would say easy for you, how easy it has become once you know the characteristics of the virus sequence data, and you can select and develop a vector-based vaccine by using this genetically engineered technology. And then you can have that vaccine which will be more homologous and more specific. Certain, just to make my case stronger for homologous vaccine, I'll just try to put some benefits and challenges. So I think anybody who is going to manufacture it or use it, he has to understand what benefits the homologous vaccine carries. Obviously, this targeted immunity, very specific, and you have to decide which one you want to use in which area, which country, which region. So it's easier in the sense of focusing it and controlling disease more rapidly. Once you have this approach, the spread of disease like those influenza, IV will be reduced. Thank you. Okay. Other interesting thing is the cost effectiveness. Obviously, basically this is point more for the vaccine producer for the production purposes. So it is cost effective to manufacture such a vaccine. Long-term immunity concept, again, it is once it's homologous and we have a booster given to the birds, So you can manage this titer for a longer period of time, because every time you're giving a booster of the same intensity, same structural components, you will have a better prolonged immunity. Emergency disease management, still in many countries, in European countries, America, their companies are making autogenous vaccine, and they name it autogenous vaccine just to cover it, differentiate from the other vaccine. Purpose is to isolate from a form organism, characterize it, and same companies propagate it and develop the vaccine. We do the same in Pakistan as well. And this, the point is, you need to register those vaccines before you market it. So in some countries, they have given permission, already permission. In case of emergency, you can market it and give it to those farms which are affected or interested. But you cannot market it or internationally sell it. That's kind of criteria, differentiate between a requirement. Challenges. I'm not going to peak. Sorry. I'm not used to speaking in house. Okay. Challenges are basically the strain specific. Strain specific means that if there's a mutation, there's a new evolution of organism, you have to continuously monitor that change and modify your vaccine accordingly. It's not going to be a simple thing, just import it from somewhere, multiply it and sell it. You have responsibility becomes more. You may call it a challenge, but this is in fact a challenge. Vaccine failure is a challenge because the storage condition, the life of that organism, studies will be very short duration. People would not have worked in detailed way upon an organism or vaccine. So it is kind of a challenge to maintain good quality vaccine with a prolonged the period, storage period, temperature. So all this information may be kind of a challenging component. The cost of production, obviously, the live vaccine will be more expensive to produce and manage. Like if you're going to do vector-based vaccine, you need a system of liquid nitrogen containers and all that stuff. So that's kind of a challenging part. Now, just to share with you our experience when I was doing job at NRC, Representative Research Concept. I have worked there for 35 years as a chief scientist, virologist. So that's my experience of developing homologous vaccines and controlling some diseases. So I'm going to share with you two events. First thing, 1988, we had an outbreak of adenovirus 4, which was later on identified which type it is, adenovirus. We did not have the facilities, nor the typing facilities. Sequencing was not popular in those days, and other criteria were dim, but we were able to identify that it is adenovirus. We isolated it, grew, and used it basically in the early stage in birds. Inoculated it, collected the liver material from the bird. There we see the DMs, we purified material and used this antigen as a vaccine. It really worked miraculously. The whole thing, as I said, started in 1987, 88. And I always say, should I say the word luckily, or maybe you can say bad luck? No, luckily we did not have any vaccine available in the market. So we had no option but to try something. So I think that was the first experience that we basically purified it and used it as a vaccine, homologous vaccine. And fantastic results. We were able to control this disease in broilers. It was spread initially in broilers. In later years, it also transferred to the breeders. And we had that problem for a few years. In the meantime, we developed cell culture-based vaccine in 1994, 95. And we were basically comfortable using this for many years. So the disease was quite controlled, very rarely few occasions, you would say. And then after a gap of many years, later we found the emergence of new adenovirus. And as I said earlier, there's no vaccine of adenovirus 2, no vaccine of adenovirus 11, 8, yes, there's a vaccine. So we had to again a challenge to develop those vaccines. And luckily we have done it, developed it, commercially it's available in Pakistan, manufactured here. So we are trying to handle this using this idea, this approach. So shortage of time, I cannot get into detail how, but you just have a feeling that this technology, this approach, we have used it successfully under our conditions. Second example is avian influenza virus. In 1995, Pakistan had first outbreak of H7N3. Same situation, you can imagine 1995, the what would have been our status. So, and again, luckily, I don't know, but there was no vaccine available for H7 in those days. So we had again same challenge, but this time we're more kind of equipped, more knowledgeable after experiencing the adenovirus. Luckily, this outbreak occurred in Murray, just 100 kilometers from here. There's a mountainous area, and the breeding stocks were mostly raised over there because of the temperature issues, because the close housing was not introduced at that time. So we decided amongst us that we have to develop the vaccine, use that, isolate, grew it, inactivated it, and use it. And just to share with you, just for the thought-provoking process, many organizations, including FAO and OIE, they resisted and they basically asked us to stop it, don't do it, suggested us. They refused to sponsor any funding or research. No, you should not be doing this. You will be in trouble, whatever. But the point is, based on our own experience, we did it. Luckily, we were again successful. 6 months time, disease was controlled there. And you can see H7 outbreak, next outbreak occurred late somewhere in 2004. The next outbreak of H7, which was again reported from Karachi South, not in the north, new entities, but by that time we were, more, you can say, equipped and more knowledgeable. So we could manage it. Within 1 year, we were able to control it. Same approach, same virus. Now we were a little bit more economically better. So we had this, we developed, first time developed in the country, policy of this disease control. National Contingency Plan was developed first time in 2005. How would we encounter it in case this happens with H5? Because in those days, H5 was also coming up. So we devised a strategy, and it was the main theme was that we'll have a vaccination plan whenever there's a strain, and we will be giving compensation to the people who vaccinate and report. And when they report, the culling team will destroy those birds which are positive and give them compensation. And I'm pleased to share that out of 300 million rupees in those days, we spent only 60 million rupees, Pakistani rupees, to manage all this detail. And detail of H5 is already mentioned over here. So we had the first outbreak of H5N1 2006. I mentioned the number of outbreaks and all the records. So you can see slowly it was going down, and this strategy somehow worked. And I'm really proud to mention that since 2008, we do not have a single report of H5N1. We did have a few H5N8 outbreaks, very isolated, they were contained. We used H5N8 vaccines same day. And today I met a number of delegates coming and meetings, and people are always skeptical that maybe we are hiding something. I'm sorry, that's what I feel. But as a matter of fact, we should feel proud of it, that we have used an approach which is scientific, and some people did not agree at that time. But interestingly, now both OIE and USDA, they are supporting the use of vaccine as one of the major solutions for influenza. So it's okay. But I think we were right in those days, and we are right today. So similarly, anything which we come across now in our country, it's our responsibility to study it and encounter it and design your strategies. The strategies used by somebody else may not work in your situation because of the common practices difference, because of strain difference, because of whatever. So it needs a continuous research, continuous development of new vaccines, which I think country like our country, we need approach of vaccine. We cannot compensate and pay back everybody, killing every flock and then paying back this money to the farm. That's not because of our own situations. So anyway, the good thing to share was that Pakistan is free. And another thing to add is that we opted for a carpet vaccination. In the beginning, it was the idea was organized a kind of it's called zoning approach of vaccination. There's a problem. I'm just done one more slide. Thank you. There's a zones are made, and you do vaccination around the zones, and then you treat the bird inside in a different way. We use water-based vaccine, now used by others. They use oil-based vaccine. What's battery-based vaccine? Adjuvanted vaccine, aluminum hydroxide, but fantastically in our state. So using those approaches, it was possible to contain it. That's why I'm explaining is that I still believe that our problems, our viruses, our organism, we have to understand them more. More we understand, more comfortably we can devise the control strategy. So just a summary, what was the outcome of our past, experiences. I just, I think, said most of the things I just mentioned it. I think it led us to develop a vaccination system in the country. Before that, we did not have any private sector involved in vaccine production in this country. So that basically provoked. Now there's 4 companies in Pakistan. They both went through registration of vaccination. Few of them switched to the pharmaceutical, but few are still working with vaccines. So that's, I think, part of the contribution that we will become self-sufficient in producing the vaccine in the country. Another benefit is that in spite of flock culling approach, we were able to get recognized that this approach can also be workable, and we saved a lot of money from the farmer. Not only money, but the other word, the hate of the farmer as well, because you are the killer of the flock. So we are living a different culture. So we have to understand that. I think this also led us to have the first national reference laboratory in the country. So that was our success. Before that, poultry was not recognized as an entity for research purposes. So I think that's a part of this effort. We were able to convince the government officials of contribution, and we were given money to establish a setup. And obviously, it resulted in controlling of the disease. And I think we are, colleagues are struggling, thinking of using the similar approach in other diseases as well. This is the last slide I have. Just tried to summarize. Somebody came late maybe. I think we still believe that avian pathogens are continuously monitoring, continuously mutating to survive. This is the bar of survival of the fittest. So we'll continue having these challenges in future as well. As we know that in current situation, despite using different vaccines in our country against different diseases, we still face the challenges. So I can say with confidence that efficacy of number of vaccines used is becoming questionable on account of the sequence data and serological data, which now we have about those organisms that circulate in our country. And we also should recognize that simply culling or improving the biosecurity alone cannot help to control these kind of infectious problems. We have to see all these things together. So it's not one-line solution. You cull all the flock and you will control the disease. Not happening. Vaccine matching has been helpful in the past, and it could be a major tool in future vaccine development. As we heard yesterday, our colleagues talking about different approaches to develop those homologous vaccines. And obviously, homologous vaccine by using the circulating organism and applying it, it's a better approach to better control the infection. And this approach may need to be more specific to the region or countries. I cannot use the virus from Italy. They cannot use the virus from Pakistan. Once you understand, why would you use this? So same way, we are also very important, as other countries are very important. They don't want the virus. So if we want our virus, then we are also important. So I think that's the thing I just want to say. And the last thing would be that we need to strengthen our monitoring system in the country of virus isolation, characterization, developing new, because the industry, the sector which is producing the vaccine, they cannot take that much research independently. So we need to have a more connectivity, enhancing the efforts of those researchers and institutions to bring up new molecules, new strains, new seed, so that new vaccine could be developed. And I think that's lost. Thank you very much for patience.